Sure. Here we go.
Okay, so I'll begin by introducing Isaiah. He has earned a master's degree in clinical microbiology and is currently working on his PhD in the conservation sciences program at the University of Minnesota, and is a graduate fellow in the Minnesota Aquatic Invasive Species Research Center. And also hot off the press, Isaiah has recently been hired as the leader of the Minnesota Department of Natural Resources Fish Health Lab. So he's wearing a couple of hats today and is going to be presenting on his work with koi herpes virus. Take it away, Isaiah.
All right, so thank you for that introduction. I'm going to be talking about one aspect of my dissertational work. This project is entitled Investigation of Koi Herpes Virus Disease Periods and Factors Influencing Transmission. So first of all, before I get into koi herpes virus, let me talk about common carp. So they're important for kind of two different reasons, one of which being that they're extremely important for global food security. I pulled this chart here on the lower left-hand corner that's just showing some of the more common aquaculture species worldwide. We kind of deal more with the catfish and rainbow trout here in the United States, but as you can see, the growth and just kind of overall sort of tons of production of tilapia and common carp in the green is much higher. So these have been really exponentially growing worldwide. So it's not something that we really see here. There's not much of a market for common carp in the United States, so it's not really like something we're exposed to. But worldwide, they're extremely important for global food security. Also, koi carp is a color variety of common carp, is extremely important to the hobbyist industry. So it's almost crazy to call it a hobby with how much money people spend on these fish. I've spoken to one person individually who told me that he bought a koi for $18,000 and that it kind of ruined his marriage. But a lot of people invest like heavily in these koi fish, and it's an important industry worldwide. This is something that's practiced, breeding and selling these fish worldwide. And they're also culturally important in a lot of countries. So one of my favorite examples is how carp are like, they swim up this river and jump over a bridge and turn into dragons. And it's a Chinese legend. And if you graduate with your PhD or make some other big milestone, it's kind of they use the same word for the graduation, is basically your carp that turned into a dragon. So at any rate, they're really important culturally. They were introduced into North America for the same reason of global food security. They were sort of hoping to kind of just put them into a lot of different water bodies. So the predecessor to the U.S. Fish and Wildlife Service introduced them because people wanted to have them around for food. But that kind of brings us to the next part of why carp are important, which is that in the United States, Australia, New Zealand, they're actually an invasive species. So we've identified pretty severe environmental issues that they cause, and there's economic effects of that. There's millions of dollars that are spent trying to reduce carp populations in order to improve water quality throughout the United States. And they're one of the worst aquatic invasive species in terms of just sort of the amount of places that they've been introduced and the amount of habitat destruction that they cause. So this USGS clustered specimen records is just kind of showing where the distribution of carp is in the United States, and they've virtually moved into every freshwater that they possibly can access. And they can occur in really dense populations where they're basically 90% of the biomass of fish in a water body. So those are the two reasons why common carp are important. And then diseases of carp are ones that affect global food production or could possibly be of interest for a pathogen biocontrol strategy, are interesting for that reason too. So it's important to understand koi herpes virus, which is an important pathogen of common carp, leads still these days. It's still a probably the number one limit factor of common carp production worldwide. And it's really the only pathogen that's really been sort of identified as a potential biocontrol tool for common carp. That's not something I'm going to focus a lot on today, but if you have questions about that, please feel free to reach out to me. It's a double-stranded DNA virus that has a it uses the sort of like membrane blubbing, so that has part of the membrane on the outside of it as well. It causes sort of a kind of hemorrhagic disease. So you can see like hemorrhages throughout the skin and in the gills of fish that are infected. Gill was previously seen sort of as like the entryway for KHV, but it's more likely the skin that KHV enters through, and then gill is like the secondary site of infection. And you see like these really bad hemorrhages and necrosis in the gills, which along with some neurological symptoms might be what actually kills the fish. In the United States, KHV was introduced likely in New York through the koi trade is what we're thinking it got introduced, but it's pretty widespread. So these black dots on this map are showing all the places where there's been outbreaks of KHV in wild populations of carp. So this doesn't even count places where KHV has been identified, like in someone's koi. These are all the places where a wild population of carp has had a mass die-off and it's been identified. A lot of my first year, my PhD was just kind of trying to map out where these outbreaks occurred in Minnesota and surrounding states. So that's the black box here. We also found carp edema virus with bees. It's another carp-specific virus. And sometimes in co-infections with KHV. So that was pretty interesting. But there's definitely a lot of these outbreaks that occurred in Minnesota, and we were able to isolate a strain after doing this work. So what I want to talk about for what I want to talk about for the rest of this talk is one particular question that we were interested in, which is to kind of ask the question of what is the mechanism of KHV transmission and what are some factors that are involved in that. We already know in general that the KHV seems to be really specific to carp. There's really not great evidence of any vectors of disease. So like, another like arthropod vector or something that concentrates the virus, or reservoirs of disease like waterfowl. There's one study that was pretty recent where they found KHV DNA in waterfowl, but haven't actually shown like transmission from like waterfowl to other carp, or even demonstrated that the virus they found was viable. So it could just be DNA or something like that. So there's no evidence of that yet. Not that it's impossible, but it doesn't seem to be the primary way. Also, it's possible that KHV may be like sort of sequestered by invertebrates, and that might be like another source of KHV transmission in the wild, or that there could be like sort of mechanical vectors of transmission. So other fish species that come into contact with KHV and they're able to transmit it. Most of those studies that have demonstrated that have been like in recirculating aquaculture systems. And so they might not be reflective of what actually happens in a wild population of carp. So that kind of leaves us with two different mechanisms of transmission, one being direct transmission, where fish are actually touching each other, and indirect transmission, where like a sick fish is shedding KHV into the water column, and then the water acts as like a vector that can move the virus around. And that's pretty important for like disease modeling strategies, and kind of like how you deal with like patches and a network and whatnot. But this is just sort of a basic question that hasn't really been answered. Which one of these two, or what combination of them is the mechanism of KHV transmission? We do know that transmission of KHV in wild populations seems to happen more often during the mating period. So this is actually a picture of one large female carp, and she's full of eggs. And these male carp will actually develop what's called nuptial tubercles. So they'll get like kind of like these like hard like sort of nodules on their skin, and they'll lose some of the mucosal layer, and they have like a more rough texture, and it actually lets them during the breeding period come into really close contact and like press their heads up against the female carp. So there's a lot of like kind of vigorous like rough contact that's going on, and this is interesting because we see a lot of the transmission happening right during the breeding period, and we also know the skin might be the main entryway of KHV, but that the mucosal layer can disrupt that transmission. So having this direct contact, where you're kind of brushing away that mucosal layer, you have physical contact between these fish, seems to be a really great way for KHV to be transmitted. So that gives us some clues as to what might be the main mechanism. There are also other factors that may come into play. So some obvious ones that I wanted to look at were viral load. So how much does viral load increase transmissibility? How much does contact rate increase transmissibility or transmission? And also periods of disease. So when you think about, when you're sick with a cold, you have this period of disease where it's called like a prodromal period, where you're not sick, and you don't feel sick and you don't have any symptoms, and you go about your day normally. But then once you start experiencing symptoms, then you might act differently. You might stay home, you might call in sick to work, and your behavior will change. So this has been studied with other animals, but it's possible that that behavior changes pretty significantly. And we know that there are even neurological symptoms with KHV that could impact behavior. So we looked at comparing the clinical and subclinical periods of disease. So the first trial that we did for determining periods of koi herpes virus disease in juvenile carp was pretty simple. I took 16 juvenile common carp. So these aren't like very small common carp. They're about 60 grams to like 100 grams. So not quite adults, but they're more robust than like the, little larval or like juvenile carp. They're, like a few grams. And that's important because you can get like peracute infections in them where they just die off really quickly. But we would give them sort of like a unique marker. It's basically this little injectable elastomer that you can put a few little colored spots on their face and use that as like a color code for identifying each fish individually. And then inoculate them with a low dose, it's 100 TCID 50 per milliliter of KHV on a swab. And so what we're doing here is we're putting it on the caudal fin so we can kind of disrupt the mucosal layer, make sure that they get infected, but sort of know where the infection was. It's not immersion where it could show up anywhere, including the gills. And then we can use the gills as a secondary site of infection to determine when they actually start shedding the virus. So by exposing them and then waiting and screening them each day with this gill swab, we could determine the latent period, which this latent period that we determined is pretty similar to the literature. If you look around at the temperature window that we use, 22 to 24 degrees Celsius, which is the optimal temperature for KHV, this is pretty standard. And then there's also a period that hasn't been measured in the literature to as far as I know, which is the prodromal period. And this is the time between when you become infectious and when you start showing symptoms. And this was about 4 days, give or take a day. And then when I should say that these symptoms that we're identifying, these are visual symptoms that they're pretty common in the literature. We used basically some darkening of the skin and loss of that mucosal layer. It's kind of the first thing that you see when these fish get sick with KHV. And then as they progress through the symptomatic period, this is, after those first disease signs are noted, to whatever endpoint we're using. I used moribundity as an endpoint, and I defined that as fish that were basically laying in lateral recumbency, or ones that, were not really avoiding the net. And all of the fish reached that point in this trial. So it's possible that some of them could have survived, and that would have increased the infectious period, so the amount of time it took for them where, but they wouldn't have been prematurely killed. So this infectious period is a couple days lower than like the average that I see at the same temperature in the literature, but it's pretty close. So that gives us sort of this incubation period of subclinical disease, and then a symptomatic period that we compare in the next trial. This is just a quick look at the viral load. So this is from those gill swabs of these fish that we were infecting. And it's kind of, the error bars are pretty big, but what we're looking at here is you can see kind of an obvious little peak here at 5 days post exposure, and then you start to see some of those first clinical signs. And then there's another peak at 9 to 10 days post exposure, and then fish start to die at this point. So this is, not the full sample of fish once we get down to here. So that might be part of the reason why it's tailing off pretty abruptly. disease trial setup. This is a little complicated, but I hope I can explain it in a way that everyone gets it. Basically, what we're looking at here is I had two different groups of carp. One group is these vector carp, or V.carp we'll call them, and these are the ones that we expose to KHV. So we do it the same way. We give them an individual unique marker, give them inoculation by caudal fin swab, and then wait 2 days, which is the average length of the latent period, and we confirm whether they were infected or not by swabbing the gills. And if anything was determined not to be infected, we just remove it from the trial. We ended up with 12 fish for each replicate that we did, and then the rest of them we would leave for our V.carp group. Then we had a second group of what we call susceptible or sentinel S.carp. These ones, we have 15 per group, and we have 8 groups for the 8 trial replicates that we did. And on each day of the trial, we'd move one of these vector carp into a really large tank. So this is like swimming pool size tank with pretty quick flow through. So 1 tank volume per hour. And I want to say that the reason for doing this large tank volume with this large volume of water is because I'm trying to simulate something more close to what we'd see in the natural environment, and kind of move away from this like recirculating, like, 30-gallon aquarium setup that I see in a lot of other trials that, might be artificially increasing the amount of indirect transmission that you'd see just from water acting as a vector. And move more towards what you would see as a normal stocking density. So this stocking density that we get here is kind of the optimal stocking density for carp and aquaculture systems, and it is it's still a few orders of magnitude different from what you'd see in a wild system, but it's probably more comparable than anything else in the literature at this point. And so we'd move in one of these vector carp into this tank, and then there'd be 15 susceptible carp, and we'd let them incubate together for 24 hours. We did this under 2 different conditions. So trial 2A, all 8 replicates were done with the vector carp being loose in the tank, and it can interact freely, have direct contact with the S.carp. And then in trial 2B, all 8 replicates happen with the vector carp enclosed in a double-walled cage, so there's just water sharing. So theoretically, what we've got here is we have indirect and direct transmission both happening in trial 2A, and only indirect transmission happening, or water sharing happening, in trial 2B. So again, back to 24 hours of incubation, and then during the trial 2A, we'd actually look at these fish and watch how many times this vector fish came into contact and like touched these susceptible carp. And we'd watch them for 15-minute intervals 6 times during the observation, during this 24-hour period. After the 24-hour period is over, we take out the vector carp and take out tissues, gill and kidney, to quantify viral load. While the susceptible carp, we move them into individual tanks, so 15 individual tanks, so that we don't get tertiary infections. And after 4 days, we can determine the viral load from their gill and kidney, which will give us the number of fish that were actually secondary infections, so the ones that got sick, and that gives us our transmission rate for each trial replicate. So in total, we do 8 trial replicates of this whole thing under direct and indirect conditions. And the replicates are 4 replicates during the subclinical period and 4 during the clinical period of disease, based on our first disease period trial. See here. Lost my mouse somehow. All right, here we go. So the first thing that we were trying to figure out with this trial was sort of that overall mechanism of transmission. What's the primary mechanism of transmission? So this first chart here is showing a force plot. So we look at like the condition of like which fish we're comparing. So this is the contact trial versus the indirect contact trial. So 2A versus 2B, focusing on the contact trial. And then what we have here on the x-axis is the odds ratio for transmission. So this is the odds of transmission happening under that condition. And the dot shows you where ratio. If it's further towards the right, that means it's a positive odds ratio. The odds are in favor of that happening. If it's on the other side, the odds are not in favor of that happening. And then this line here is showing the 95% confidence interval. So the way to read this is that if this line crosses the red dotted line or 0, then it's not a significant odds ratio. And these are basically mirror images. So this is the contact trial 2A versus trial 2B, and this is trial 2B versus 2A. And you can see they're mirror images. So the odds of transmission were much higher and significantly higher than under the scenario where we don't have any contact and we just have water sharing. So that gives us kind of our overall answer. Yeah, it's direct contact that is probably the more important source of transmission. And not only that, but if we look at just the contact trial and we compare the subclinical period to the clinical period, we can also see that that transmission was significantly higher during the subclinical period than during the clinical period. So that early portion of disease where there's no disease signs is when most of the transmission happens. And we see sort of the opposite. It's not significant odds ratios, but for the trial 2B, so under water sharing only, you see kind of the opposite relationships. The early period of disease, there's no evidence of transmission, and then we see a little bit of transmission during the late period of disease. And then each of these is just showing each day or each replicate of the trial individually. So you can see how during the early 4 replicates of the trial, there's a lot of direct transmission happening, and direct transmission is significant. The odds of direct transmission is significantly higher than indirect. And then we kind of see the line get blurred here. So from days 10 to 12, the odds of transmission under direct contact is not significantly different than the odds of indirect transmission. So indirect transmission becomes more important during this late period of disease. Just kind of looking at this another way, if we look at this y-axis, this is the number of secondary infections, and then the x-axis is the days post-exposure for the vector carp. And you can see the same thing here with there's a lot of transmission happening during the early period of disease, during under contact, and then it starts to drop down. And then in the indirect contact or water sharing trial, we see that there's no transmission here, and we get it just a little bit towards the end. So this kind of gives us our overall direct transmission seems to be the primary mechanism of KHV transmission. This chart here is a correlation plot. So for trial A and trial 2B. So the factors that we're looking at here, just to kind of start teasing apart which factors influence transmission. We have days post-exposure. We have the viral load in the susceptible carp after we check that. Then we have the number of secondary infections or the transmission rate here. And then the viral load in the vector carp, which we'd expect to be positively correlated with time. And then this is the contact rate, so the number of contacts that the vector carp makes with susceptible carp per hour. And then this is a vector disease score. So this is kind of scoring based on 4 categories of disease. For instance, we look at fin fraying, and that gets a score of 1 to 4 with a total of 16 points that they can get for all 4 categories of disease. And that's just kind of getting gross pathology quantified, so we can look at correlations. And then this final box here, I'll explain a little bit more in the next slide, but this is basically transmissibility, which is the probability of transmission given contact. And a few things that are interesting to notice here is that the number of secondary infections, we already talked about this, but the number of secondary infections is negatively correlated with time under the contact trial. But we do see that the viral load goes up, and if we were to look at a viral load plot similar to the first trial, it looks very similar with a kind of a peak at 9 and 10 days post-exposure. But what's interesting here is that we actually see that contact rate is decreasing over time and is actually negatively correlated with viral load. So as viral load is going up, the contact rate is going down. See the same thing with contact rate versus disease score. So basically, as these fish are getting sicker, they're not making as much contact with the sick fish is not making as much contact with the rest of them. So that's kind of interesting, and it kind of influences how we almost get like kind of like a negative correlation with secondary infections and viral load. I should have started this out by saying that ochre color is negative correlation and purple is positive correlation, and these asterisks are just pointing out which ones are significant, have significant p-values. For trial 2B, the number of secondary infections is positively correlated with the viral load, which is more what you would expect. So basically, viral load is definitely additive here. And if we look at this in like a linear model, which I'm not going to show here, viral load is actually additive in the direct contact trial as well. So it's not really, there's a negative correlation seen here, but it's probably more related to this contact rate decreasing as the disease gets worse. So kind of going into this transmissibility concept, this is just the raw data from the trial. So what we have here is the different days post-exposure. And then we can see in trial 2A, this is the number of secondary infections we have. In trial 2B, number of secondary infections. And we also have the average number of contacts per 15-minute period that we saw in trial 2A. So to kind of tease apart transmissibility, we can kind of divide this number of transmissions that actually happened by the amount of time that was available for those to happen. And we get the probability of transmission given contact when you have both direct and indirect transmission happening. Because remember, this one, trial 2A, both direct and indirect transmission are happening in that tank. But since we know that we have one trial where we're looking at only the indirect transmission, if we subtract those, we can actually look at subtract, the transmission that's not attributable to direct contact from transmission attributable to both direct and indirect transmission. Then we can calculate the transmissibility due to direct contact separately from transmissibility not attributable to direct contact or indirect transmission. And what's interesting about this is we can see how this transmissibility for direct contact is dropping off. So it's actually KHV is less transmissible by direct contact as the trial moves forward, and even at let's see which one here. Yeah, at 10 days post-exposure, transmissibility by indirect contact is actually higher compared to direct transmission. And then kind of like a more, I find this easier to think about it this way, but if we were to calculate this transmissibility of due to direct contact in terms of sort of like minutes of contact that are necessary for KHV to be transmitted, this is maybe a little bit more like easy to look at, but we have like a half an hour of direct contact is necessary for KHV to be transmitted earlier on during the first 4 days, which is the subclinical period of disease. And if we compare that to the clinical period of disease, with the exception of the 10 days post-exposure, we see that it's much lower. So just like 4, 8, or 6 minutes of direct contact is necessary, and with an average of about a half an hour during the early period of disease, and less than just about like 7 minutes during the late period of disease. So the reason behind this looks to be that the contact rate is much lower during the late period of disease. So to explain why there's much more transmission by direct contact, well, that makes sense if there's a lot of contact going on. And why transmission seems to drop off in the late period of disease is explained by this really sharp drop off in contact. So this is a violin plot showing contacts per hour in a control group in the 4 replicates of the subclinical period and in the 4 replicates of the clinical period here. Taken as a group, the subclinical period is no different from the control. So this is, counting contacts between an uninfected carp that's marked with all the other fish in the tank. And there's no difference between this taken as a group and that. At 5 and 6 days post-exposure, the contact rate is significantly higher and lower compared to the control group, but taken as a whole group, it's not different. But if we compare the subclinical period to the clinical period, or the clinical period to the control group, we can see that the clinical period is a lot lower contact rate. And not only that, but the type of contact also changes. So the little letters down here are indicating the type of contact that we categorize. So if it's an S, it's a social contact, which means that all the fish are basically they're facing the same direction, they're shoaling together, and they seem to kind of like, come into close contact for a little bit and they'll move apart, just like they do in the control group. But we start to see some of this aggressive contact towards the end of the subclinical period and always during the clinical period. And what this looks like is the susceptible carp, the ones that are uninfected, start acting aggressively towards the infected carp. And this is pretty interesting because what we would see is that infected carp starts to move away from the group or swim in a different direction, different orientation. So it's not shoaling properly with the group, and starts to get kind of chased off when it gets close. And towards the end, we actually see that carp, when the neurological symptoms really set in, it's just swimming listlessly through the tank, and it'll sometimes make incidental contact with the rest of the S carp group, and sometimes just be chased off. And virtually all of the contacts we observed were due to that chasing behavior towards the end. So this is the main conclusions of this study are, first of all, that the primary mechanism of KHV transmission is direct contact, not necessarily indirect transmission with water acting as a vector, and that direct transmission of KHV is driven by social contact and viral load. So I didn't demonstrate the viral load part here very clearly, just because I didn't want to show you a big table of linear models, but basically viral load is also additive with contact rate. Social contact occurs primarily during the asymptomatic period of disease. This is really interesting to me because it's maybe showing like an empirical example of a mechanism that drives virulence to transmission trade-off. If you're really, really virulent and you make your host sick immediately, then you might not have like this great time where your viral load is adequate for transmission to happen, but your host is acting totally normal and is coming to contact the way that it normally should. But as the symptoms start to set in, then that individual is going to act very differently, and maybe even elicit some sort of like social avoidance from the rest of the group. Disease avoidance hasn't been demonstrated very often in the literature for fishes, but there are a few examples that are really cool out there. One really awesome example is a study, can't remember the author right now, but they actually look at tadpoles and show that really elegantly, they show that the disease avoidance behavior of these uninfected tadpoles exposed to infected ones is actually from chemoreception. So they're detecting something in the water and moving away from them. It's not visual or something else like that. And then finally, indirect transmission is more likely to occur during the late stage of disease when there's a high viral load. I did quantify viral load in the water column, but the way that I was doing the quantification, I couldn't tell any difference between early and late stage of disease. It seemed to just be KHV was always there. But I want to point out again that this is a pretty low stocking density trial, but still it's orders of magnitude higher stocking density than what occurs in a natural environment. So indirect transmission might not play as important of a role in transmission of KHV. And with that, I'd just like to thank everyone who's involved in this project, the bigger project that encompasses this one. Lots of techs in the field and in the lab, and Dr. Moore and Dr. Phelps for kind of helping me orchestrate this entire thing, and also for our funding from the Environmental and Natural Resources Trust Fund and through MACERC. And with that, if we have time for questions now, I can answer some questions now, and also feel free to reach out to me and ask me any questions you might have.
All right, thank you very much, Isaiah. Great presentation. Lots of good stuff in there, and we've got time for questions. So Jim, I see your hands raised.
Yeah, thank you. Hey, Isaiah, that was a very elegant and a well done study and a nice presentation. And also congratulations on your new job.
Thank you.
One of the things about herpes virus is persistent infections after recovery. And I don't think your study addressed that, but can you give us some thought as to what the risk then of those surviving fish would be for reactivation of infection and then exposure of susceptibles. So that would be a long-term study and clearly beyond the scope of your work. But could you kind of address that issue as to what level of risk there is in those persistently infected fish? And does temperature or other stressors actually control that for some periods of time?
Yeah, that's like the question. I think that's great. I think that the interesting thing about these populations that get exposed to KHV is that you're going to have those long-term survivors, and that's going to have some really interesting demographic effects in a population that's been exposed. You're going to end up with this long-term sort of infected population of adult carp that are mounting immune responses to KHV. They're not necessarily getting disease signs anymore, and they also don't shed the virus as much as they would during acute infection. So you're going to see, I think that maybe this subacute period of disease and the behavior that you see there and the type of transmission and transmissibility you see there is probably more similar to what you'd see in the natural environment. So you basically need to have lots of close contact in order for transmission to occur. And the only way that transmission is going to happen is if you have naive fish in the population, which are going to be young adult carp that aren't necessarily exposed to the virus in a way where they develop immunity to it or long-term immunity to it, that are coming into contact with this reservoir group. Another thing that's interesting too is that we see that the antibody level increases in these adult carp that are exposed. Basically, the larger the fish is, the more antibodies it has. And so that might mean that they might stop shedding at some point. They might not necessarily continue to act as a reservoir over time, and they might, they might not even necessarily shed it anymore or completely clear it possibly. So.
Great. Dave, I see your hand up too.
Yeah, I dropped it into the chat box, but this is nice work. First question, have you published it, or where have you published it?
It's not published yet. We're working on that. It's close. We have it drafted and we have a journal picked out. We just need to.
Okay, I think this will open a lot of people's eyes, and it's good information. The second question sort of relates to what Jim brought up here. Given that effectively carp can recover and become reservoirs for KHV, how did you start off and determine KHV naive fish?
Yeah. So the way that we did that is I basically had 400 fish in a tank, and I just sample enough of them to determine that they're all negative. So we take about 20 or 30 fish out of that, take out all their tissues, test everything individually by qPCR and see if there's any that are infected. They were supposed to be coming from a disease-free source as well. So it's kind of like two lines of defense there, but there is always that like lingering possibility, but I think that we're pretty sure that these are disease-free as far as we can determine. So qPCR is pretty sensitive, and we're usually able to tell. So
great. Chris Phipps, how about you?
Hey, Isaiah, that was a really nice presentation. Really good work there. You sort of answered part of my question. I was thinking a lot about the water and some of those, the more those fish are in the tank, the more potentially more virus is there. You said that you did test the water, but you weren't able to quantify it, or you weren't able to tell the difference between, say, time zero or later times. What kind of tests did you use?
So basically I did kind of like the basic eDNA test. So you use like I was using a 0.1 micrometer nitrocellulose filter, and then I would run
like a known volume, or
5 liters of water through it, and then digest the entire filter in a microbiome kit, and then try to use PCR to quantify that. But what I found was just that they're all about the same. There's no significant difference in them, which I think is not necessarily due to there being the same amount of KHV in the water. I think it might have more to do with just that the test not necessarily being very sensitive to that difference. So yeah, it's that some of that work is tricky, like ultracentrifugation or electropositive. I think I can't remember if it's electropositive or electronegative that you need. Opposite of what I had. If you use something that has an opposite charge to the virus, then you can actually like get those like virions to stick to that.
You can pull it out,
yeah, and concentrate it better that way. And then also it's eDNA is just sort of a messy technique. Like from what at least in my hands, it's messy. It's gets out of the filter and you're constantly bleaching everything and hoping that you don't get cross-contamination. So.
Yeah, it can be challenging for sure.
Yeah.
What do you think it's related to this? And because you alluded to the idea that in your closed system, maybe the virus is going to be much higher than what's out in the wild. I mean, has anybody else looked at water going, just rivers and streams or lakes or whatever, looking at viral load just out in nature? Has that been done? And how might that relate to what happens in an experimental system?
Yeah, so there's one really good one. I think there might be two good papers. One of them's by Miwa from like 2014, I think. But I might be getting that wrong. But at any rate, he went out and did I think electropositive membrane filtration and quantified KHV DNA from a location that he kind of knew was a spawning location. And they could detect it. It was a lot lower than what I found.
Okay. Yeah.
And also the other thing to point out about that is that in a natural environment, like in normal water, KHV is not viable for very long. So some studies show that it's
another question.
matter of hours or minutes that it's getting preyed on by bacteria and protozoans and whatnot. And also no one's actually demonstrated that is infective after they take it out of the water column. So
cool. All right.
Could just be DNA contamination. We don't know.
Right. It's there, but is it viable? Yeah.
A little bit of it is. That's what this study shows.
Right.
At least a little bit of it is, but I just can't see that necessarily contributing in a really great way. Though, if you think about like patches of carp that are infected, if you have even a little bit of transmission from one patch to another, that could have some significant impacts on like moving KHV around the landscape.
Right.
So.
Cool. Thank you.
Yeah.
Great. Isaiah, you got a question here in the chat box from Heather. Do you think that carp that are symptomatic with KHV interact less because they are stressed and maybe tend to isolate themselves?
This is
spread the virus. They spread the virus when they are asymptomatic and are socially interactive.
Yeah, I think that's what's going on one level. There's another thing. I don't know why I can't remember names of authors right now, but there's an amazing paper that demonstrates behavioral fever in carp. So actually, carp will, when they get sick, or a lot of fish when they get sick, they'll move to warmer water, and it like helps them sort of like activate their immune system. So it's a way of kind of like you could imagine like a sick fish with any pathogen might be moving towards that warm water, while all the other fish are going to stay in water that's more comfortable, optimal, maybe moving towards higher oxygenated water, while the sick ones are moving towards heat. And they actually showed that KHV has this crazy I think it's TNF alpha homolog receptor that it produces, and it binds to that signal that's kind of producing inflammation, making these fish move to warm water. So there's kind of like this delay effect where the virus is trying to keep those fish from doing that weird behavior and activating their immune system, and also potentially keeping them closer to new susceptible hosts. So I think that's a pretty fascinating idea, but behavioral fever might also be part of the reason why they move away. So they might be just kind of like seeking warmer water, or there's also the neurological impact that I was talking about. So a lot of these carp, like they'll start swimming very weirdly, and they'll kind of, you can tell like their muscles don't work quite the same as they did before they were sick. And also they'll kind of be laying on their side. And I think some cool histological studies have shown that the parts of the brain that are impacted by KHV are some of the ones that are associated with shoaling behavior. So that shoaling behavior might get disrupted by just those parts of the brain being degraded. And so.
All right. I don't see any more questions. I'll give you just a second for someone to jump in. Okay, thank you very much, Isaiah. Virtual round of applause for you and an excellent talk.